last time we talked about the muscle tissue organization. One thing we talked about was the fascicles, right? These lines that you could see, right? That would also indicate the line of pull, right? So that if they were all kind of going in this direction, when the muscles contracted, right, they'd pull it up in a, toward the origin over here, right? They pull that arm up, right? So these are your anterior delts. They pull it up forward. Here's your pec muscles, right? They pull it up over toward this way, right? Or maybe they pull a little bit downward on this side right here, right? So those fascicles would also indicate the action, but remember what those fascicles were, right? We talked about the organization of the muscle tissue. So when we looked deep inside, right, this piece of flank steak right here, you can see these lines, these individual lines were those fascicles, right? And what those fascicles were, right, this is the lines right here. When you looked at it from a microscopic point of view, each of these lines right here, under the microscope, were one of these right here, right? These fascicles. So each fascicle had those individual cells right here, right? These are the muscle cells, right? And that's what we're going to be looking at today, the muscle cells. And then we're going to be looking inside this cell right here, right? So this is, electron microscopy. This is very small. Each of these is about the width of a hair or so, right? Or a little bit smaller, each of these muscle cells. And we're going to be looking deep inside. So we're going very deep inside the muscle today to look at the microanatomy of the muscle, right? So we're looking at a little bit about the properties of muscle tissue in general, smooth and hard as well. And then we're going to be looking deep inside, looking around the muscle cell itself, and then inside the cells, like what is special about these muscle cells that are going to allow these properties right here. And then we'll look deeper inside, right, the actual organelles and into the sarcomeres, right, the actual things that are going to cause the contraction. So real quick, because we are going to be covering cardiac and smooth in a separate lecture, but while we're talking about muscle tissue and its properties, all these properties that I'm going to talk about apply to not only skeletal muscle, which we're focused on for the most part, but also cardiac and smooth, right? All these muscles, all these ones, what they do is contract, right? They all contract. They all have some kind of excitability, right? They all have extensibility. They all have elasticity. So let's look at what these terms are. The contractility is your key property of muscle tissue, right? It's unique to muscle tissue in that here's a muscle cell. And when it does shorten, right, it's going to be connected to others in such a way that it shortens and it pulls on things, right? This is a smooth muscle, which might be arranged in sheets. This is a skeletal muscle, which is ultimately attached to tendons, which is attached to the bones right there, right? So this is your key property of muscle tissue and the unique thing that no other tissue has, right? The other property right here is excitability, right? This is also shared with your nervous system, but your muscles in response, your cardiac muscles, your skeletal muscles in response to stimulation by the nervous system, for instance, or hormonal, right, is going to create a wave of excitation down the length of it, right? So here is a spinal cord. Here is the excitable nerve tissue, a red and purple neuron that sends its axon out the spinal nerve and then connects to the muscle fibers, right? So there's an excitable tissue here, and then these are excitable too. So that when these cells send off a signal, right, it's going to cause these cells to have a signal, right? And we're going to talk about a lot about what these signals are and what they do to cause the muscle to contract, actually. Okay, we'll see that again. Signal going down, and then there's a signal that's going to be going along there, right? They're excitable. There's something about the membrane here, which we'll talk about a little later, right? That allows it to pass this wave of electricity down the length of the fiber. All right. And so the other ones, that's, contractility is kind of key. Excitability is another one, but it's not unique to muscle tissue. And then the other two, extensibility, right over here, relates to contractility. That is, it can be stretched, right? Just like you can stretch your whole muscle. And when it does stretch, it's going to be able to contract over a range of lengths right there, right? So this is related to its contractility part, but it can do it at a range of lengths, which we'll talk about as we get into the microanatomy next time, right? And then the other thing is there's some elasticity built into the muscle tissue, itself, as well as along the connective tissue that forms the muscle tissue, that helps organize the muscle tissue. So elasticity, right? Just like that kind of elastic fibers, right? You stretch it and it rebounds to its resting length after a contraction or a stretch. So those are the other two properties, and we'll touch upon those as we get deeper into the anatomy part. But these are the four properties of skeletal or muscle tissue in general. So when I talked about contractility, what I mean is that the muscles are shortening, and the whole muscle is going to shorten, right? And that shortening is going to be due to contraction of the myofibers or muscle fibers, the cells that make up the muscle tissue, because those muscle cells themselves are going to get shorter. And the fact that they get shorter is due to these contractile proteins that we're looking at right here within the muscle cells themselves, right? So your muscles are filled with muscle fibers, which contract. The muscle fibers are filled with these contractile proteins, which are going to contract, right? So when a whole bunch of these contractile proteins down the length of the muscle cell contract, it's going to, if there's enough of them contracting, right, it's going to shorten, belly's going to bulge up over here and then create a pulling force. Okay. And so what we will see is that, what's happening inside here at a micro level, right over here with the proteins that are involved in contraction, is there's going to be these little kind of ratchets right over here. And that'll be the contractile components of it, the sarcomeres, as we'll see. And then there'll also be some elastic components within the muscle tissue as well as the connective tissue, right? But this is a sort of schematic of what is we're going to see how that works today, right? Because these little things over here are the actual ratchets, right? And then whenever these kind of slide up against each other, get shorter, that's going to cause a overall shortening. So you go from a relaxed state to a contracted state. And here we go, we're flexing at the elbow right there because your muscles pulled on here. Right? So that's what we're going to be looking at today. Now first, let's look at looking at the individual muscle organization again. We had the whole muscle, which had a bunch of fascicles within it. Right? And within the fascicles over here, you had packed with muscle cells, right? So here, in its entirety, is a fascicle. And then within it, you have 1, 2, 3, 4, 5, 6, 7, 8 muscle fibers or muscle cells. I'll be using these words interchangeably. Usually I just call them fibers or sometimes myofibers, right? But they're the actual muscle cell. One clue as to whether you're looking at the muscle cell, because this kind of pictures can get confusing, right? Because it's a tube within a tube within a tube, and then you get these little things in there. Look for this green dot right there. That's the nucleus, right? In this slice, there's a nucleus, right? And that'll tell you you're looking at a single cell, right? So you got 1, 2, 3, 4, 5, 6, 7, 8 cells in this picture right here, okay? Within a fascicle. And it's just a cartoon, doesn't mean there's 8 cells. Remember that other picture of the rabbit? There was like 20 or 30 of them, and there could be a lot more. Okay, here is a person. Over here, here's his arm, there's his muscle. You got a bunch of muscles. Maybe this is a foot long. Maybe that's the size of some of the longer muscles right over there as far as scale goes, right? Some of them might be only the size of a shrew or a chicken egg. Sometimes they're quail eggs. Sometimes they're earthworms, right? As far as size goes. Right, so we're looking at the scale going deeper and deeper. And just for reference, some point, if you want to know, these are about the width of your skin over here, right? Your thin or thick skin right over here. And we're looking, we can still recognize things by the human eye without a microscope, a grain of salt. Amoeba, and right about here, the width of a human hair might be about the size of a bigger muscle cell. Okay, so that's about the scale we're looking at when we're thinking of those fascicles that are packed with a bunch of these. Right, so you wouldn't be able to see that from your skin, or if you remove the skin and we're looking at muscle, but when they're all packed together, then you can see the fascicle. So that's, they're huge cells, right? Here's a skin cell, much smaller. Right, so they're really long cells. They might not be that thick, but they're pretty thick. They don't have one in here, so that's the best I can use. And then just for comparison, there's a skin cell. We're going to look deeper down. It's about the size of a coronavirus right there, that HIV virus. And here, over here is a cell membrane. Over here, so again, going back when we were looking later, the cell membrane would be covering this whole thing, right? Tiny little thing relative. But going back over here, pretty sure when I first learned this, I learned about this size right here. This is roughly the size of one of the microfilaments that are packed inside the muscle fiber. Right there, some of those proteins, some of the contractile proteins. Right, so they're small compared to, your hair right there. There's a whole bunch of those packed in there. But then consider, how big they are compared to, this compared to your cell membrane over here. Okay, so that's just for your scale purposes. And so when we're talking today, it's just get an idea, right? This you can see the muscle cells themselves, they're microscopic, although you can see some of the larger ones because they're width of hair, right? But we're going to look inside that width of hair, right? For all these rods that are running alongside within the muscle cell itself, right? So here's a cross section of a muscle fiber, right? There's the fascicle. Here's the muscle cell. Here's a bunch of fascicles. Here's a fascicle, and then here's an individual cell. Okay. And so the first thing you want to note is remember those connective tissue covers. You have the endomysium covering the individual cell. And within that endomysium, you'll see things like blood vessels, like the capillary. Right here, that's going to be supplying the oxygen and nutrients right to the muscle cell because these need a lot of oxygen and nutrients. Right. You'll also have running through here, this little yellow thing here, which is the axon of a nerve fiber, right? Because they're going to need stimulation by the nervous system to contract. So those are both within the endomysium. Just deep to the endomysium is your cell membrane itself, right? In red here. Right? So you got the cell membrane called the sarcolemma. Over here, the specialized cell membrane that's going to be excitable. And then that's just deep to your endomysium. And then within here, right, you're going to have a nuclei over here, right? Because this is you're gone from outside the cell to the cell membrane. Now we're looking at inside the cell, and we have the cell nucleus. And one thing you might notice, there's one nucleus here and another one here, which means there's actually a whole bunch of nuclei in any given muscle cell. There could be hundreds of nuclei in a muscle cell. And that's important because these can be over like a foot long, really long cells. And to get all the proteins that it needs, you need nuclei that are going to make those protein. So muscle cells, skeletal muscle cells are multi-nucleated, right? They run all down the length. You could have hundreds of them in a single muscle cell. Okay, so there's two of them right there. Now what else we got? Oh, so that's the nuclei of the cell. And you'll also notice there's this thing over here. This is another cell that's sitting within the endomysium. And we'll talk about this later, so I'll put a pin in that. But these are the muscle stem cells, right? The muscle stem cells of the muscle, which means when there is a breach of some sort, there's some kind of damage, or maybe the muscle wants to grow in response to exercise, these stem cells will start dividing. And what they'll actually do is incorporate themselves into the muscle cell to help build it. They don't make new muscle cells, they just add themselves into there. And we'll talk more about those when we talk about muscle repair and muscle during exercise. But you can forget about those myosatellite cells for now. All right, so nucleus inside the cell membrane, and then we'll also see mitochondria over here. Right? Mitochondria, what do mitochondria do? They make ATP. Muscle cells need a lot of ATP, especially if they're those ATP type ones, right? So there's a lot of mitochondria in there, right? Making a lot of energy. There is also within the sarcoplasm, all the red things in here. And this is where we're going to be getting to the center of our talk here, these myofibrils. Right, so you had a tubes, the fascicles within it. Those fascicles were filled with the muscle cells, another tube. And then within that smallest tube, you'll have not really tubes so much, but more like rods, right? So they're packed with all these rods in it, right? Going all the way down the length of the muscle fiber, right? So most of your sarcoplasm is taken up by these red dots, which are your myofibrils. Right? This is a myofiber, the cell. These are myofibrils. These myofibrils, in turn, we're going to look deeper into what they're made out of. These are made of myofilaments, muscle proteins. When you see the word filaments, it usually means a long protein. And these are very long proteins, right? Strings of proteins. So the myofibrils are going to be composed in a certain arrangement of myofilaments. So let's look at that levels of organization again. Right? We had the muscle over here, which you can see, the fascicles, which you could see because they're bundles of myofibers, right here, all these stripy things within a fascicle. And so those are the myofibers. And those myofibers, right, which is the cell, is packed with these myofibrils, which is an organelle, myofibrils. The myofibrils are made up of a certain arrangement of myofilaments. Right? In particular, these are actin and myosin filaments, protein filaments. Right? So this is the levels of organization. You can guess like some kind of question like this on the test, right? So here is one of those myofibrils, right? Most of your muscle fiber, myofiber, is made up of myofibrils, which again is composed of myofilaments, which we'll look at in a second. Other things that are important within this that we'll need to talk about here are going to relate to the whole contraction. So we want to make sure we understand what these are, although we will go over them again. Right? This is just kind of a primer. We're going to talk about these in detail, but for right now, you have the sarcoplasm, right, packed with the myofibrils. That's making up most. There's also a lot of mitochondria for energy and myoglobin. Myoglobin is like a hemoglobin, which carries oxygen, but it's for the muscle. That way, we have a kind of supply of oxygen within here. In fact, that's what makes muscles so red besides the blood vessels, right? Myoglobin. And those myofibrils, right, which are in the sarcoplasm, the structure is thick and thin myofilaments. Again, we're going to look at those. They're running all down that long muscle fiber. And their function is muscle contraction. This is what's actually going to contract, get smaller, cause the muscle to contract. Another structure here in purple over here is the sarcoplasmic reticulum. This is the muscle version of an endoplasmic reticulum, if you remember what that was. right? This membranous bag. In this case, the important part is it's surrounding the myofibrils. It's a membrane complex, and its function is to store and release calcium ions, which are going to be important in the contraction. Sarcoplasmic reticulum, that's these purple things right over here. Again, we're going to get more into detail about them. The cell membrane itself, instead of just being called the cell membrane, it's got its special name, is your sarcolemma, specialized for excitability. And it's also going to be containing the neuromuscular junction. That is where the nervous system meets the muscle and tells it to do things. And that's your sarcolemma. And then these green things over here, again, we're going to cover these more in detail, is the T tubules. And the sarcolemma kind of folds in over here, this excitable membrane. It kind of invaginates. You can see the little holes right here. They're extensions or invaginations of the sarcolemma to go deep inside the muscle cell. So if this is excitable, so are those T tubules. And they're going to form a connection, a structure with those sarcoplasmic reticulum over here, called the triad, right, which is going to connect the excitability of the cell to the release of calcium. Again, we'll go over this in detail. So let's just put a pin in that, and we can talk to it when we get about that. It'll be next week when we actually talk about the actual functions, when we get the physiology of it. But I want to just introduce that now, and then we'll talk about those later. All right. So here is our levels of organization. Muscle. Here's a fascicle filled with these things right over here, right? And you can see little stripy things over here, right? The stripy things down here, very consistent on a muscle cell. You'll see what are called striations, right? That is, they have this stripe pattern of dark and light bands, right? And then what we'll see is that these dark and light bands are going to reflect the actual arrangement of your myofilaments. And that's kind of what we'll be talking about today. So here's an actual histology picture, a close-up of 1, 2, 3, 4, 5, 6, 7, longitudinal sections of 7 myofibers. Here's the nuclei of the fibers. And then you see these little stripes over here going down them, right? Little stripes. Those are called striations. That's why skeletal muscle and cardiac muscles is considered striated. So we're going to see what these light and dark patterns mean at the molecular level and what they mean, how we relate them to it. So here again, stripe patterns, dark band, light band, dark band, light band, right? Going all the way down the length of the cell right here. And what we're going to see is that these myofibrils, right, running down the length of them is what's going to make this light and dark pattern. So it's pretty amazing that all these are going to be due to these individual ones on each myofibril, but they're all lined up together within a muscle cell to make this overall clean appearance of light and dark patterns right here. Dark bands, light bands, dark bands, light bands. Right. So that's what these striations are. So when we look deeper into it, we'll see that these dark and light bands are due to arrangements of these thick and thin myofilaments that make up the myofibrils within a myofiber, right? So here's an electron, a very close-up picture over here, a photograph. Here is a light band, here's a dark band, light band, dark band, right? So this is what we're seeing all the way when you look at a single muscle, right? They're all lined up together to form these dark and light bands. And what we'll be really kind of looking at is a more cartoon type figure where we're going to see what those dark and light bands, what's the significance of them, right? Because they're going to make up these red and green myofilaments, right? That are going to be the actual contractile units. Okay. So this is what is known as a sarcomere, right? So the sarcomere and these dark and light bands created by the organization right here goes from Z line to Z line. here to here. This is a single sarcomere. And so it starts at one Z-line, ends at the other one, right? And so your light band has half a light band on either side of this dark band, really within a single sarcomere. In the middle, right down the middle of the sarcomere, you have what's called an M line. Right here, an M line, right? This goes down the middle. It'll kind of form the basis, we'll see, of these myosin filaments. Okay, so your light band, right, which is half a light band is on either side of these Z lines. Over here consists, for our purposes, of these thin filaments, the actin filaments. Okay, actin filaments, and they're all the red ones, which you can't see too good because these green ones are taking place. The green ones are tightened, but we're not going to worry about that just yet. Right, but they're going off the Z line right here. And that's your thin actin filaments. So then you have your A band over here, which is these purple bands right here, which are your myosin. I don't have a purple marker, but there are these right here, right? These are the thick filaments. So I'll alternately call these thick or thin filaments or myosin and actin. Same thing. Those are just the particular protein names, and they're particular types. We can get more detail, but we'll just call them myosin and actin. Right. So one way to remember the I band is the light band. There's an I in light, and there's a band. There's an A in dark. Right. If you want to kind of remember A band, I band. So these are often called these were historical purposes. These names are I band and A band, but you can also call them light band, dark band. But those are two, and that's what they consist of. You'll notice that the A band does not have myosin filament in it, but your I band does not have myosin filaments in it, but your A band has myosin and actin filament overlap. Okay, so your A band, for most of it, has both actin and myosin. There's one region that doesn't have any actin filament in it, and this is called the H band or H zone. And that's the zone. This is important to remember because this H zone or H band is the zone of non-overlap. That is, in most of the A band, you have actin overlapping myosin, but in this H band right here, there's no overlap here. So it's clear it's only myosin in the M line. It's basically on either side of the M line right here. So we're with me so far about the structure here, the Z band or Z line, M line in the middle of the sarcomere. Sorry, the Z band, Z lines are making up the sarcomere. It flanks, that's basically the definition of the sarcomere. In the middle of it, you got this M line. And then you have a half a light band, full dark band, and then another half a light band here. Right? The light band on either side is all actin and titin, but we're not concerned about that right now. The A band has, for most of it, with the exception of the H zone, an overlap between actin and myosin. Right? So whether you totally get this right now, you'll want to understand all these elements at some point. Again, I should have had this in the beginning, right? But the sarcomere itself is going to go from here to here. So when we look at the contraction, how this is going to, the sliding filament theory, we're going to look at how this sarcomere is going to get shorter, right? We'll see this kind of collapse and move in from Z line to Z line, basically, is what we're going to see. All right. So the contractile unit is that sarcomere, and the contraction of the muscle is due to contraction of the fibers, right? The contraction of the fibers due to contraction of the myofibrils. A sarcomere is just a section of the myofibril, and that's considered the functional unit, right? So just to kind of get that clear, right? If this red pen here was a sarcomere, right? This blue one was another sarcomere. This black one was another sarcomere. Here's another sarcomere, right? All this together makes up the myofibril, right? Just a series, right, of these a series of these sarcomeres make up the myofibril. And they're not in any way independent units, right? I'm just using this as an analogy, right? These are functional units. It's all one big interconnected structure. But so each one of these colors is an individual sarcomere. And when you line them up end on end like that, you have a myofibril, right? And each one of these independently, or not independently, but all together, they're going to be contracting each one of these, right? All the way down the length of this at once right there. So sarcomere is just a section of the myofibril. All right, so let's look again at what's going to happen during this contraction phase, right? We already took the cartoon version of the actual picture. Here's the Z line, right? Here's the other Z line over here, and here's the light and dark bands, which are going to be composed of those things, right? Light bands is mostly just actin filaments. Dark bands had some overlap, except in the middle around the M line right here was the zone of non-overlap, right? And then when the muscle cell contracts, when the myofibrils contract, right, we're going to see that these red ones, the actin ones, are going to slide over the green ones. And what we'll see is they're going to, because of the way they're attached, they're going to pull it in, right? So this whole thing is going to go from this to this, all the way down the length of the muscle fiber. Okay, so we're going to get more into depth in this right here, right? So here is one section. Here's that Z disc. Z disc, this is just looking at one side of it. Here is the M line over here. And then here are the thin filaments in red, thick filaments in blue right here. And again, the titin, we're not going to worry about the green over here for a second. Okay, so the thin filaments, this is called the sliding filament theory, right? They're going to slide over there. And when they do, they are going to pull this Z disc toward the M line. Okay, that's what's going to be happening here, right? They're going to interact in such a way that they pull over the Z disc toward the M line on both sides and all the way down the length of that myofibril. Okay, so notice that over here, I have this line right here that's measured along the length of this actin filament. If I take that same line, I put it on the one in the contracted filament, nothing's changed. This actin filament stays the same length. Right? Your myosin filament right over here, it stays the same length. So when I'm talking about shortening of the muscle, what we're getting at here is that there's going to be a sliding over here, right? Nothing is actually shortening per se, except for the whole structural unit, right? No proteins are squishing up in any means. The best way to think about this, if you know what an expandable ladder is, right? You see them on the back top of somebody's truck, they take them out, they put them on the roof side of the house, and they expand it out. Neither of the sections of the ladder is changing, right? But they're changing relative, so they're lengthening. And then when you close it, it'll shorten, right? And that's basically what's going to be happening here. But the way it's all connected to each other is going to create this pulling force. Now looking a little bit closer, right, at a closer range here, we're going to look at this section, right? Let's see over here, actually, right, where the Z disc, Z line over here, and the actin filaments coming off it, right? So these thin filaments, your actin filaments, are directly anchored to the Z line. Right, so this is actually another type of this is actinin, right? It's a different type of actin. So they are directly connected, right? So if these move, they're pulling the Z line with it. Okay, so that's important. The thick filaments, right, they are attached to them, but that's not going to come into play during the sliding filament theory. They're attached to them through this protein called titin, Titan is nothing to do, they didn't even discover this. I think they discovered it maybe 20 years ago. They've been talking about the sliding filament theory. And this is the biggest protein discovered yet. And they didn't know it was there until they did something or other. So it's kind of funny that they had this whole mechanism and they didn't even talk about the biggest protein that ever discovered. But this Titan, hence the name, is so big, it's also attached to the Z disc. And for that, I'll just mention right now about its function, although again, it doesn't get into the whole slidingness of it. Right, this right over here, you see this kind of spring over here is stretched out, over here in the relaxed or stretched state, and then in the contracted state, it's shortened right here, right? So what this is going to do is when the muscle is stretched, right, it's because your myofiber, the muscle cells are stretched and your myofibrils are stretched. So when they stretch, you have a little bit of this stretchy protein over here that is going to allow it to recoil and rebound a little bit. So when we talk about the elasticity, the intrinsic elasticity of muscle, Titan is your main sort of protein that we're thinking of like this, right? So it's allowing this recoil, but it's also preventing overstretching of the sarcomere because it'll only stretch so far. If it overstretches too far, that would be a problem right there, because then the myosin filament couldn't interact the way it needs to with the actin filament. And so that's what you want to know about Titan for our purpose. It adds to the intrinsic elasticity of the muscle fiber. The other part will be the connective tissue cover, the connective tissue layers around there, right? That intrinsic stretchability and the extrinsic would be your connective tissue, because these kind of collagen fibers have a little bit of stretchiness to them. So they'll also allow a little bit of the elasticity. Okay. So that's it for Titan. Let's get back to the actual ones that are going to be involved in the sliding filament theory. And let's look at what happens during an actual contraction here. Right, so here is, again, that stack of sarcomeres. It goes from here to here, remember? These are the individual sarcomeres down the length of this. And these are the individual sarcomeres. And when it contracts, I'm going to ask you what happens. What can you see just looking at the myofibril itself? If you compare the sizes of these different elements, the dark and light bands, you want to ask yourself what changes and what doesn't. Right? So your dark bands, does the dark band change at all? No, good answer. It doesn't change at all, right? Does your light band change? I'm too impatient to wait, but yes.
Kind of looks like it, yeah.
Yeah, it does, right? It shortens, right? Just from a macro view, or not macro, but looking at the myofibril, you can see that when these myofibrils are going to contract, those light bands are the first thing you may notice that changes, right? But also you'll notice the Z lines. Okay, these are the Z lines, which make up sarcomere to sarcomere. You'll notice, they are going to be pulled in closer to this M line right here, right? So let's look a little bit over here and see how that works, right? So here's your I band over here, and we know that has shortened, right? We know that's going to shorten when you do this right here, right? It's gone from this length to this length on either side of it. And when that shortens, right, we see that this Z line over here, it goes from here to over here, here to over here, right? So the Z lines are being pulled toward the M line over here. Does the M line change at all in this given sarcomere in this picture? No. Does the A band change? We already said no, right? It's still in line with each other, at least for a single sarcomere, what you're talking about, right? So you can draw this out, draw one A band with the M line and draw another one directly in it, underneath it, and you could draw the exact same thing. And then for your Z disc, those will be what changes right here. So what else changes over here? Right? The A band hasn't changed, but remember we have this H zone, the zone of non-overlap. It's worth being clear about is I know it's sometimes called band and zone. Don't worry about it. It's a zone of non-overlap, right? The H zone. So that's where actin and myosin don't overlap, right? And here there was a big H zone, and over here, right, when during the contraction, that H zone has collapsed, right? Because these actin filaments have moved toward the M line, right? Increasing the overlap, right? So the H zone, which is zone of non-overlap, decreases, right? It shortens. Okay, are you everybody clear on what, well, everybody clear on how it could happen? If you have this picture with you during the exam, right? If you have a picture like this, you could answer any question, right? What happens? What happens to the Z line? Does it pull toward the M line, right? Yes. What happens to the I band? Does it contract or does it not change, right? Oh, it contracts, obviously. Just looking at this picture, right? So a picture is worth 1,000 words. One good thing to do is maybe draw it, right? Draw this picture right here. All right, so and here's from your book. Over here, this is drawing 2 sarcomeres. Over here, right, same things, right? Your I band, right, on either side, the 2 halves of an I band on either side of the Z discs. And then you have the A band, right? And over here, they're showing it a relaxed muscle, partially contracted muscle. Over here, right, when the zone of the H zone decreases a little bit, right? And a fully contracted muscle, that H zone will completely disappear, right? Go fully contracted over here, right? And pull past there. So that will be the maximum contraction right here, right? So you could do this over a range of length and get a really strong contraction right here until it can't shorten anymore. These are all the structural units, right? This is what's ultimately causing your contraction of the muscle. Okay, so when you look at something like this individual muscle cell, right, and you know there's thousands of these little things over here inside a muscle fiber, because they're packed with those thick and thin filaments that are going to make up those stripy patterns right here. When it contracts, right, that's what's causing this bulging of the muscle, like all those muscle cells within it are contracting, causing the whole thing to contract, right? And they're going to pull on things, right? That's going to create a pulling force on the bones that it's attached to. So here it is again, right? Here's the muscle cell. There's the nuclei. This is an individual muscle cell packed with these myofibrils, right? Here's a string of myofibrils together, right? So when it contracts, it's getting shorter. And so at an individual level here, right, these will contract all along the length of the muscle cell, right? Pulling. They'll all do it at once too. They'll all shorten all the way down the length of it, right? Want to see that again, right? Contraction, right, is due to all those contracting at once. And then if we look at that, right, that contraction over here, right, here are the individual sarcomeres, 1, 2, 3, 4 sarcomeres over here. And when they get the signal to contract, right, they're all going to increase the overlap, right, pull all those Z discs closer to their individual M lines and cause the whole thing to shorten, right? So contraction of the whole myofibril down the length due to this whole contraction, each individual sarcomere down the length of that. And then those are the elements that we want to know right here, right? Z discs, right? And the actin filaments overlaid by the myosin filaments, which are attached to the M line. And then the H zone and the I bands and the A bands. All right, and that's just the anatomy of it right here. That's all we're going to really go over today, right? But you know, when we're doing all these movements, when you're seeing your muscle bulge like that, right, it's due to thousands, probably millions of these little ratchets like there that are going to kind of contract and then make all your muscle cells contract, which is going to make the whole muscle contract. All right, and then next lecture, we're actually going to get a little bit deeper into, you know, the interaction between the actin and myosin filament that actually causes the so-called sliding filament, right? And what the role of calcium, what the role of the sarcoplasmic reticulum and the T-tubules, and eventually we'll get into the neuromuscular junction, right? So we're going to look at the sequence of events that actually causes this contraction next time.